Okay, so our next speaker is Melissa or Missy Monson. Oh, there she is. And Melissa was a postdoc with Sue a couple of years ago and is now at ARS and at the National Animal Disease Center here in Ames. So Melissa, looking forward to your presentation.
Well, thank you very much for giving me the opportunity to come to speak today. And I'm going to talk a little bit about connecting turkey immune responses to food safety and intervention strategies against Salmonella. As Jack mentioned, since I left ISU, I have joined the USDA Agricultural Research Service as part of the Food Safety and Enteric Pathogens Research Unit, which is located at the National Animal Disease Center, which is part of the National Centers for Animal Health. If you haven't figured out that the federal government likes acronyms, I'm going to try to avoid them for you all. But this is a picture of the big building. Our campus is out on the eastern edge of Ames, and my labs. If you could go through the building, my lab's basically on the first floor on that side. So today I'm going to talk on a smaller scale of projects here. I've been with the USDA just 3 years, so I'm just going to highlight 2 of the things that we're working on. I work in the food safety unit, which does research in swine, cattle, both dairy and beef, turkeys, and a little bit of chicken. My projects and focus are typically on turkey. But before I talk about science, it is a tradition for doing presentations with Sue to start your presentation with a fun fact. And so I still need to give my fun fact for Sue. And this Hui-Jun stole the words out of my mouth. I was going to say that Sue always went around and told us that she had the best lab group in the world. And I would say that that is directly due to Sue being an amazing mentor and all the support and guidance that she has given us. I'm not a very good photographer. I realized I did not take enough photographic evidence, but I really enjoyed my time with Sue as a postdoc and working with her large dynamic group. We had a lot of fun. These are just some snapshots of us at conferences, at Sue's house, at different locations in Kildy. Here's that same mural that Hui-Jun just showed, for example. And we did a lot together and had a good time doing this. And just as proof that we did in fact work, not just pose for photos, here are a couple of pictures from some of the animal trials from my postdoc. For example, this some of the my partners in crime here are in the audience working on avian pathogenic E. coli or avian influenza. You can see our differences in some PPE requirements there. And these are Sue's favorites here, the Leghorn and Fayumi chicks. And thank you for John and Hui-Jun, Jang, that took those photos of the birds. So after my postdoc, I've moved back into turkeys and have been working at the USDA location here in town at the National Animal Disease Center, doing work as part of the food safety unit, looking at interactions between the food safety pathogens, their host, the other commensal microbes, and the environment. So our goal is to basically disentangle these for all the different major food safety pathogens in our livestock species. I have the background in poultry for our unit, so my work has been primarily in turkeys and looking at Salmonella. So I probably don't have to really spend a lot of time with this audience talking about why Salmonella is a public health concern and an issue, but very briefly, the CDC estimates that there's over 1 million cases of this each year, leading to over 400 deaths. And based on 2019 numbers, they estimate that there's over $4 billion in costs associated with salmonellosis in humans, a large portion of which are from foodborne sources. And the reason that this is such a challenging issue is multifactorial, but some of the big ones are the fact that our livestock species are typically asymptomatic carriers of salmonella. So it is not a pathogen for the turkeys, but it causes a big issue if it makes it through the production system and into human food. And as another challenge, salmonella is highly diverse. There are over 2,600 serovars, making it difficult to have interventions that are applicable across this diversity of strains. And we also estimate that there's approximately 16% of strains out there that are resistant to antibiotics, many of which are now becoming multi-drug resistant, and this makes it a challenge therapeutically to be able to control infections when they happen. So in the context of these challenges, the Food Safety and Enteric Pathogens Research Unit, or FSEP as we call it, spends a lot of our focus on developing and evaluating interventions against salmonella. And it's a range of different things that we've investigated: live attenuated vaccines, modulation of the gut microbiota, non-antibiotic feed additives or immunomodulators, and also environmental controls such as antimicrobial peptides or biocides. The couple figures I'm showing here actually work from my unit before I started. So they've been working on a large number of things, some of which are a little newer, so we don't have published work yet. Since I've started, I've been helping on projects related to all three of the interventions shown here at the top. For time today, I'm going to focus in on just one of our projects looking at responses and interactions for a live attenuated vaccine. So for this project, we were focused in on looking at vaccination against multi-drug resistant Salmonella enterica serovar Redding, or Salmonella Redding. And we are interested in this particular serogroup or serovar of Salmonella because of its importance to the turkey industry. In 2017 to 2019, Salmonella Redding was associated with a widespread outbreak and linked back to turkey meat and turkey products. This outbreak generated over 400 cases and was spread across 42 US states and also Canada. So it was really a broad issue. And in the after effects of this, the poultry industry and the turkey industry in Iowa especially were very interested in understanding whether the vaccines that we have available are able to actually control against Redding. Can we use them if Redding becomes increases in prevalence and becomes an issue again for the potential to kind of work against the potential of another outbreak? So for this project, we received some funding from US Poultry and Egg to look at vaccination in the context of Redding in Turkey. And the idea here is if the vaccines are effective against the serovar, we could prevent another outbreak. But the challenge here is that the vaccines, live attenuated vaccines or Salmonella, are typically based off a Salmonella typhimurium backbone, and Redding is a different serovar from this. So the question was whether this was going to be protective and effective against this other serovar. So for this work, we utilized the only commercially approved vaccine for use in turkeys in the United States, the Elanco Avapromegan egg, and a ARS developed and patented vaccine called BBS 866 that I need to give a shout out to Sean and Brad Beerson, like some of my colleagues in ARS who developed this vaccine. So we tested this in turkeys, and alongside the vaccine work, I also looked at the host response, characterizing the interactions with challenge and vaccination using transcriptomics in the cecal tonsil from these turkeys. And we were interested in the cecal tonsil as an important site for intestinal mucosal immunity and understanding how the immune system is going to interact with Salmonella redding. So this is just a outline of our study here. The only things I really wanted to mention is, along with the different vaccines that we gave, we did our vaccination both by an oral gavage, but also aerosol and water to kind of mimic what is more done in industry. So we don't have the nice sprayer that they use and it's used commercially. So instead we were misting the birds individually to do our aerosol vaccination, and then they were boosted by drinking water. And at 7 weeks of age, then we challenged and collected tissues at subsequent time points. So this is just a subset of the results here, kind of representative of what we are seeing. The plots are showing tissue colonization by redding in cecal tonsil or spleen. And at 2 days, the Salmonella has colonized the cecal tonsil, but not made it to the spleen yet. So it takes more than 2 days, but sometimes between 2 days and 7 days, it was able to disseminate out to the spleen. The situation for the effect of vaccination isn't quite as strong at 70 days post inoculation, but by 14 days, it's clear that in all of the vaccinated groups, we're able to reduce and or clear Salmonella in these birds. So the reductions here were up to 3 logs. So to make a long story short from the transcriptomic work, essentially the responses to redding were only observed at that early acute 2 days post-inoculation time point. The effects here were primarily decreases in expression and mostly observed when we compared the redding challenged animals to the controls. There's very minimal changes or almost no changes in those that were vaccinated. And interestingly, rather than big shifts in a lot of immunologically related genes, although there are a few up here for the vaccinated animals, the redding challenge compared to control didn't seem to impact the immune system very much, which was interesting, as we expected it to for the cecal tonsil, but maybe we have some tolerance mechanisms going on, and maybe 2 days is actually still too late. So there's still a lot of questions here. But what is interesting is that redding had an impact in reducing expression of nearly 40 different types of families of intestinal transporters. The examples I've got here are a sodium ion transporter and fatty acid bile transporter. There's also nutrient transporters, lots of things involved in those mechanisms, as well as some tight junction proteins. And it's maybe a little bit of a chicken versus egg question here. Sorry for the bad pun. That when vaccinated, these expression changes aren't observed. So potentially, if these gene expression changes don't happen when the birds have been vaccinated, then the negative impacts that reducing intestinal integrity by decreasing tight junction proteins could not having those changes could potentially prevent colonization. On the other hand, if there's less colonization, there could be less impact on the host. So there's still some mechanistic order that we need to investigate here. So in summary of this part, both vaccines were cross-protective in turkeys against Salmonella redding. And I did want to plug quickly that the BBS 866 vaccine that ARS developed is a DIVA vaccine, which means it is differentiatable of a vaccinated animal compared to an infected animal, which is really important for surveillance and something that the industry and the Food and Safety and Inspection Service are talking about a lot right now. And on the transcriptomic side, the changes that we're seeing in the cecal tonsil could potentially reflect some disruptions in intestinal integrity and barrier functions that could have something to do with the very successful colonization of redding in turkeys. And ultimately, we still need to better understand exactly when, where, and what cells are most important to control of the amount of Salmonella colonization that is happening in these birds. And for that reason, as a first step toward that, this can segue into my second project, which is trying to gain a kind of background baseline understanding of immune cell types in turkeys so that we can go back and better investigate the interactions with foodborne pathogens like Salmonella. And for this work, we chose to, again, continue with transcriptomics, but in this case, look at gene expression patterns of specific immune cell types. And since immunological reagents for turkeys are limited, we chose to do this through single cell transcriptomics. And the benefit of this method is that it allows you to capture gene expression of individual cells. So you have an idea of the profile of cell A, cell B, cell C, and then you can take that profile and use biological knowledge of the gene function to identify which cell type those cells belong to. And this doesn't require any turkey immune specific reagents. And once we have this understanding of the marker profiles of turkey immune cells, we can then use these and leverage them to understand specific immune responses to immunological challenges, such as with Salmonella. And for this preliminary project, we chose to investigate the immune cell expression patterns in an easy sample to work with, PBMC, so proliferative blood mononuclear cells. And we also did this in comparison to white blood cells so that we could keep the heterophils. And I'm not going to talk us through all of the details of the methods for this. So just trust me, at the end of the pipeline, like everyone talking about bioinformatics, you can't in a presentation, just trust me. At the end of the pipeline here, we get a plot that we call a UMAP, and what these are showing is each dot on these represents a different cell. So every cell is a dot, and every dot that has the same color and number on my pictures here represents clusters of cells that have similar expression profiles. And if these cells that are clustered together have similar expression profiles, they're also likely to be the same cell type. So in PBMCs here on the left, I have 14 different clusters, and in the white blood cells on the right, I have 18. Even before we went any further, this made sense because we're expecting to gain all those heterophils, which are the avian equivalent for neutrophils, in the white blood cell prep. So next step, now that we have these clusters, was to identify which types of cells we were recovering. And to do this, I use a marker panel. It's actually about 42 genes, not just the handful I'm showing here, to essentially identify major cell types based on canonical marker genes that we know which cells they're associated with based on prior literature. Just as examples of these plots are, again, UMAPs, and the purple colors are those clusters that are highly expressing the gene, and the gray is all the other clusters that are not. As examples, CD3E is a classic T cell marker, and we have a number of clusters that pull up CD3E, CD79B for B cells, CSF1 receptor for monocytes, and also a little bit of heterophils, and MMP9, which is classic for the heterophils. So based on this marker panel, we can then go back and annotate the clusters that we observed for each of these sample sets. The majority of the cells are predicted to be thrombocytes, which actually makes sense. They're the most abundant, and we didn't do a depletion for thrombocytes, something I might try in the future. But it looks like there might even be some innate immune-related genes in there that'll be interesting to investigate further. We also identified a large number of clusters of T cells, including a couple that we predict to be helper cells based on CD4 expression, and also a couple of clusters that are likely cytotoxic T cells and effector T cell types. And interestingly, just because science is complicated. We not only cluster our cells by cell type, but also a little by their functional profile, which exactly makes sense. We have a small cluster of cells that I just highlighted here that didn't cluster by cell type, but instead by their high expression of cell replication and division genes. And so these are highly proliferative cells, regardless of their cell type, but they'll cluster together based on their high replication state. For example, this little arm of B cells down here those are the plasma cells. And so there's essentially, there's a lot that we can learn at the big picture, but because as these point out that these clusters are still kind of top level, what we're working on right now is subclustering these to drilling down to break the cell types out further and identify novel markers that are still characteristic of specific and unique subtypes. So we've got this essentially the same picture going on here in the white blood cells. I'll just point out that here's that big cluster of heterophils that we expected to see that we gained. So in summary of this, single cell RNA-seq has revealed both canonical and novel markers for turkey immune cell types in peripheral blood. I don't have the time to talk about the details, but it's even interesting comparing the turkey genes to the couple studies in chicken, and it's the markers aren't always the same. So turkey is definitely not a chicken, and the both the PBMCs and white blood cells obtain similar clusters as we expected with similar canonical markers, but there are some differences in their cell expression profiles that we're currently investigating a little further just to see what the isolation prep and the steps that you did with the cells in the lab to prep them, see what it did to our samples. And we're already applying these technologies and immune cell markers to look at other tissues that are more of biological interest to our system and to look at cell type specific responses in the context of Salmonella, although you can apply the same idea to any other treatment or challenge of interest. So in conclusions, pre-harvest interventions such as vaccination can reduce Salmonella colonization in poultry. If we can reduce the load of Salmonella that enters the processing plant, this will work together with the post-harvest interventions that are already in place to prevent Salmonella from entering the food supply. And investigating host bacterial interactions in specific immune cell types will give us better power to understand how these are working together and how best to modulate the turkey immune system to be most effective at reducing colonization by Salmonella. And I need to close by thanking all of my colleagues at ARS, students and postdocs, the national program leaders for food safety, our collaborators at Iowa State. This is just the collaborators on these two specific projects. I've worked with many others and our other funding sources. This is about 3/4 of the people from FSET. All right. And before I close, this is a picture from the open house from the new balloon facility. And this was a great day, but far too serious. And this is maybe more reflective of Sue's group, if I can get it to stand there. This is maybe more reflective of the time in Sue's group. All right. Well, thank you.
Thank you. And we have some time for questions. So raise your hand and I'll bring you the microphone. And then you want to repeat the question?
Yes.
So we record it.
I could also just yell at him. Because I did work with Sue a long time, but I don't do turkey and chicken. But I have a question. Could you do the same thing with that single cell with the vaccinations strategy and look at what immune cell changes happen after you vaccinate the turkey?
Okay, so the question was whether we could employ single cell technologies to look at the response to vaccination of the turkeys. And yes, that is exactly our plan, is to look at vaccination, look at the challenge, look at multiple different serovars. There's so many different factors to investigate here. Nobody had been doing this in Turkey. And as Kent would know that the turkey from when I was working on turkeys during my PhD, not everything for the turkey chicken doesn't always translate. So we wanted to do kind of a baseline. So I had some good idea of what cells and what genes I might be finding before we went into the intestinal tract, where the situation is a lot more complicated, or in the response to a challenge. But yeah, that's where we're going.
What are the changes that can make? Because I don't do this, if you vaccinate, do the blood cells come?
So it will change. They haven't actually done, there's work primarily in viruses from chicken. So there's some work in Marek's disease, for example, or avioleucosis virus, that they can see responses to the pathogens. There hasn't been work in chicken yet, to my knowledge, in the context of Salmonella.
Other questions?
It's a really nice work. So I'm just wondering, for the Salmonella colonization, do you look at the cecal tonsil or the cecal content?
Okay, so the question was what for the tissue, the colonization levels for Salmonella in the Reading study, whether we looked at cecal tonsils or cecal contents, and the answer is both. I just showed for space in the slide to have it big enough. I just showed the tonsil. The tonsil in this particular experiment was actually a little higher load than in the contents, but that's not always the case. So we've looked at cecum, cecal tonsil, cecal contents, bursa. You can find it everywhere. The only place on the GI tract where it's a lot lower is if you have to go all the way out to the cloaca if you want to see a different picture.
I'm wondering, have you looked at the Salmonella shedding in the cloaca swab to see the vaccination or decrease of shedding?
Yes. So the second question was whether vaccination has an impact on fecal shedding. And we didn't do fecal shedding in this experiment, but we did do coacal swabs. We were trying to do for this particular study, we were trying to do our coacal swabs quantitatively, and the levels were low enough that we can't see a difference because we were trying to do our bacteriology to quantify. In a subsequent study, we did just prevalence positive negative, which changes how you prep it and what your limit of detection is. You get no numbers, but you have a lower limit of detection. And when we did that, we were able to basically recapitulate the positivity rate from the higher up in the gastrointestinal tract.
One last question.
This is a follow-up on your question, and it's related to not only swabbing after to see if there is more shedding, but I don't know in this case of this particular serovar, but Salmonella has the reputation of being dormant and then in the burst, and then when there is stress or other triggers, then it reactivates. So do you keep those bursts long enough to see if they challenge them with any stress and see if they can have infection again?
So the question was whether we have done an experiment essentially to look for reemergence of Salmonella prevalence and colonization at a later time. But we haven't done this in this case, and we've talked about it a lot. And it's a long time to take our birds out to market age, but it would be a great experiment thinking about transport stress, heat stress. There's a number of different things that be great experiments to do.
Okay, let's thank you.